In the endoplasmic reticulum, newly synthesized proteins must correctly fold before they can function or be transported to other locations in the cell. Proteins that fail to fold correctly are initially retained in the ER by binding to chaperone proteins. Chaperones prevent aggregation of unfolded or misfolded proteins and help steer them on pathways to fold into their correct shapes. Proteins that fail to correctly fold are eventually guided by chaperones, along with other molecules, to transporters that deliver the misfolded proteins to the cytosol to be degraded. If the ER's capacity to fold or degrade proteins is overwhelmed, misfolded proteins accumulate in the lumen. This triggers a program called the unfolded protein response, or UPR, that activates genes that increase the protein folding capacity of the ER and can also inhibit protein synthesis. The UPR consists of 3 parallel pathways initiated by sensors in the ER membrane called IRE1, PERK, and ATF6. The pathway initiated by IRE1 is remarkable. IRE1 is a transmembrane protein kinase with 2 catalytic domains on its cytosolic side, a kinase domain and a ribonuclease or RNase domain. Misfolded proteins in the ER lumen activate IRE1, triggering IRE1 molecules to oligomerize. The adjacent kinase domains of the IRE1 molecules phosphorylate one another, and the RNase activity of the oligomer becomes unmasked. The active RNase domain can now cleave a specific cytosolic mRNA molecule at 2 positions, excising an intron. The exons are joined by an RNA ligase. The spliced mRNA is then translated into an active transcription regulatory protein. The transcription regulator enters the nucleus and activates the transcription of target genes that will expand the ER, increase its protein folding capacity, and enhance degradation of misfolded proteins. Another sensor in the UPR, called PERK, is also a transmembrane kinase activated by the accumulation of misfolded proteins. Upon activation, PERK phosphorylates itself and then phosphorylates a translation initiation factor, thereby inactivating it. Inactivation of the translation initiation factor has 2 consequences. First, it inhibits overall protein synthesis in the cell. This reduces the influx of newly made protein into the ER and therefore decreases the load of proteins to be folded. Second, it increases the production of a specific transcription regulator that is, counterintuitively, preferentially translated when translation initiation factors are scarce. In the nucleus, this transcription regulator helps activate the transcription of genes encoding proteins active in the unfolded protein response. The third receptor in the UPR, called ATF6, contains a transcription regulator that will eventually be delivered to the nucleus. The accumulation of misfolded proteins signals ATF6 to be transported from the ER to the Golgi apparatus. In the Golgi membrane, ATF6 encounters proteases that cleave off its cytosolic domain, resulting in an active and free transcription regulatory protein that can migrate into the nucleus, where it helps activate the transcription of additional genes that encode proteins involved in the unfolded protein response. The relative importance of each of these 3 pathways in the unfolded protein response differs in different cell types, enabling each cell type to tailor the unfolded protein response to its particular needs. Activation of the genes in each pathway leads to the production of protein products that increase the ability to fold more proteins.